Hello, everyone, and welcome to the first ever Troy Physics Alumni Lecture Series video that we will be uploading to our YouTube playlist on the Troy Physics YouTube channel. My name is Nick Johnson, and I am the Troy Society of Physics Students President. This video was recorded at one of our club meetings, which usually this semester are held Wednesdays at 5 p.m. Our speaker today is Victoria Colvin, who is a former Troy chemistry and physics graduate from the class of 2019, and right now she's at Oregon State University studying toxicology in a PhD program. So, lastly, I should mention we did have some technical difficulties in the recording of this video in person. We had some difficulty with the audio and microphones in the room, so you may see a bit of scrambling to turn mics on and off to avoid feedback. So please excuse that. So without any further ado, I'll let Tori begin. Thank you.
Degree in chemistry and physics with a dance minor. I was also part of the Sound of the South dance line while I was at Troy, among many other clubs and organizations. And now I am at OSU, and I've actually switched fields, so now I am in toxicology. And the question I get all the time is, why did I change from physics to toxicology? Because they seem so vastly different. And the answer is that I've been always interested in all areas of science, and I noticed that I was really lacking in my background of biology. My last biology class was AP Bio in high school. So I really wanted to get more experience in that area of science. So after doing some researching online, I found toxicology, which is like the study of human health. And that interested me a lot, and so I went for it. So on the left is a picture of me in our cell culture room. And of course on the right, we're working from home. So I have my working from home buddy that sits in my lap. And the picture on the bottom is actually the lab of my first rotation. Because of COVID, we haven't been able to get a lab picture for the lab I joined. So you get a nice Halloween picture from my rotation. So just a quick explanation of how the PhD in Tox works at OSU. So OSU has incorporated this concurrent master's program starting in my year. So once I finish my courses, and in May I give a short seminar, I will actually be awarded my master's degree. This is not common in programs in all schools, but it's something that the EMT department thought was going to be really beneficial, because a lot of people, when they get to their end of their PhD and start applying for jobs, the jobs won't hire them because they don't technically have an advanced degree. And so this was kind of OSU's workaround for that. So hopefully after May, I will be a master's. And just the layout of how a PhD kind of works. So in your first year, most likely you will be focusing on your coursework and also doing rotations to identify your lab. OSU also does this a little bit differently. So starting this current year, they've actually done it to where you rotate through every faculty's lab in 1 week rotations in the first term. And then after that, you would identify 2 or 3 people that you want to do a full term rotation with. And then you're expected to pick your lab by the end of your first year. And then in your second year, it's all about being trained on the processes for the lab you pick, finishing up your coursework, and then at the end of your second year are your prelims. So every program and every school does this differently. For our department, we have to write a grant proposal for a hypothetical project that's completely unrelated to our research. And then we have to present that written proposal to our committee, present it to our committee, and then you will get grilled for about an hour on your coursework, the grant proposal you wrote, and even your current research. I plan to do that this coming fall. So hopefully I will pass that, and once I do, I will officially be a PhD candidate. So after you complete your prelims, for the next few years, you will just focus on doing research and writing publications and writing your dissertation. And most students will usually graduate in their 4th or 5th year and defend their dissertation, but it's really up to how fast your project goes and when you and your PI and your committee feel that you're ready to graduate. So what is toxicology? Basically, it is the science of safety. So, sorry, this little thing's blocking. Okay. So it's basically the study of health effects induced by chemicals or environmental stressors on humans, animals, or the environment. At its core, it encompasses biology, chemistry, medicine, and pharmacology. But it also can incorporate several other areas of science, such as engineering, bioengineering. I've even found places where physics can apply. And specifically, a lot of labs at OSU focus on environmental chemistry, cancer research, water quality testing, marine ecology, micro and nanoplastics research, and various different areas of human health, focusing on different organs of the body or focusing even on specific proteins. And toxicology usually involves research with animals, but it's moving more towards cell culture and predictive computer models. Oops. So some terminology that I'm going to be throwing around a lot is in vivo, in vitro, and in silico. So in vivo means animal studies, and pictured here are actually the 3 most common animals that we use at OSU: the rat, the zebrafish, and the daphnia. And a fun fact, the zebrafish model was actually developed by one of our most distinguished professors, Dr. Robin Tangway, who still works at OSU and has one of the most productive labs I've ever seen. Yeah, and then there's in vitro. So my lab focuses on in vitro work, which is cell culture. And there's actually a lot of new methods developed for in vitro work, such as human organs on a chip. So you basically recreate the processes of a human organ on a very small chip, which makes it really great for reducing the use of animals, as well as increasing the confidence in your cell culture work. And then lastly, an area that I'm very interested in is in silico work, which is basically predictive computer programs. So you would take all of the info we already have of all literature always from in vivo and in vitro work and create a computer program based on that. And then when you have a new chemical or stressor in the environment, you can just put it into this computer program and then it'll come out and tell you how toxic it is, how it's toxic, what part of the body's affected, or if it's just no concern at all. And that's something I really want to work towards, is working on in silico models. Okay, so getting into my specific research. So our lab focuses on the in vitro human lung model, and specifically we use primary normal human bronchial epithelial cells, or NHBEs. And these are actually really unique cells. So they're taken directly from a donor who has passed away. The cells are then frozen and sent to us, and we keep them frozen until we use them for an experiment. And most cells that you see in published papers are actually immortalized cell lines. So these were taken from a donor and then adapted so that they can basically almost indefinitely grow and expand. Our cells can't do that. So we have a limited amount of time between when we unfreeze them and when they're basically no longer viable. So the advantage of us using these primary cells is they actually are more closely related to the in vivo cells that you can find in the human lung. The immortalized cell lines, while they're great for reducing costs because you can keep using the same source for a very long time, they lose a lot of those basic in vivo properties. So the use of these primary cells is actually very important to my research. And we grow these in 2 different ways. So we do a 2D monolayer. So this is probably what you think about when you hear the word cell culture. We use a 96 well plate, and then they grow kind of in a flat surface across the bottom of each well. It's very high throughput. It takes about a week and a half to do an entire experiment. Yeah. And then we also grow them in 3D organotypic cultures. So this is a pretty new way to grow cells. And it's more useful when you use these primary cells. So instead of a 96 well plate, we use a 24 well plate. So the wells are much bigger. And there's also an insert put in there. And the cells are attached to the insert. So you first let them grow out in the 2D monolayer style, with media on top and on bottom. And then after about a week and a half, you would remove the media on the top, and that lets them grow upwards and lets them differentiate into the different cell types that you would find in the in vivo lung. So you would have basal cells, goblet cells, ciliated cells. They create mucus and everything. And it represents, or it more closely represents the in vivo lung, where you have an air surface, like when you breathe in, the air passes over the inside of your lung, and then the media represents the interactions with cells outside, like on the inside of the body. So the compounds that our lab focuses on are polycyclic aromatic hydrocarbons, or PAHs. So these are organic compounds formed during incomplete combustion processes, so diesel exhaust, tobacco smoking, bonfires or forest fires, and can even be found in the charred bits of cooked food. And these have been correlated with adverse health effects on many levels, from skin and eye irritation, immunosuppression, reproductive issues, and ultimately cancer. And the two PAHs that our lab mainly focuses on, and my thesis is worked around, are benzoapyrene and dibenzodef-p-chrysene, which are up in the right. And my thesis also focuses specifically on the metabolism of these PAHs. So PAHs and many chemicals in general, when they enter your body, they go through this two-step metabolism process. So usually this process helps to detoxify these chemicals. So they make them less toxic, more water soluble, and increases the chance that they will be excreted from the body very quickly. But sometimes this process backfires, and for PAHs, it's very common. So in step one, they will actually be bioactivated. So they become more toxic after being chemically changed by these cytochrome P450 proteins. And then in phase two, or step two of the metabolizing process, they will be detoxified by the same or different cytochrome P450s or glutathione transferases. And you can see in the top figure all of the different metabolites that BAP can be metabolized into. And what route BAP takes in the metabolism process depends on which CYP actually metabolizes it. And most of these are actually non-toxic and will be excreted really fast, but especially the epoxides and some of the quinones are actually the very toxic metabolites from BAP. And these can go on to react with the DNA in our cells to form a DNA adduct, also called a bulky lesion. So you can imagine the DNA strand, and then you just have this big old chemical that's sticking off the side of it. So obviously that's going to cause problems. This could disrupt DNA replication because the protein that takes the DNA apart to replicate it can't work around that bulky lesion. And this can lead to mitosis disruptions and apoptosis. Or it can actually lead, if the protein somehow manages to get past it, it can lead to a mutation on the DNA, which then can lead to cancer. And something really interesting about these chemicals and some other chemicals is that they can actually change the expression level of these proteins that metabolize them. So they can increase or decrease the number of proteins that are present in the cell after their exposure, which could then increase their bioactivation or increase their detoxification. It really depends on what chemical's there, which protein it interacts with, and how quickly it interacts with the DNA. So specifically for my thesis, so the goal of the beginning of my thesis is to characterize the metabolic capacity of our primary NHBE cells in 2D and 3D after exposure to these PAHs, and specifically focusing on BAP and a little bit of BBC. This project is actually one of the aims of a larger project that's funded by the NIH Superfund Research Project, which is the NIH gives an SRP grant to OSU, and then OSU identifies certain labs that falls within the scope of this grant and gives funding to those labs. So we are one of those labs. And the specific aims of the grant that's given to our lab is to first assess pH toxicity, which that's already been completed. That was completed before I got here. And then my project falls under the second aim, evaluating metabolism related to pH toxicity. And then third, which I don't know if I will be able to participate in before I graduate, is to describe mechanisms for pH toxicity. Basically, how the chemical interacts with the cell, what processes does it participate in or change, things like that. So there are a lot of methods that are going into my thesis. I won't get into the nitty-gritty details just to save time, but I'll try to give a very brief. So the two on the left have actually already been done. They were done before I joined this project, but I will incorporate a little bit of them as I go farther in my thesis. So they actually both do kind of the same thing. They both analyze gene expression via RNA. And so you take the RNA in the cell and then you reverse transcribe it to get what's called complementary DNA. And then you break that cDNA into smaller fragments, attach some sort of primer or adapter, and then you amplify it. So you sequence those fragments many, many times, and then you analyze the genes that are expressed. So with qPCR, it specifically analyzes the genes that you have a primer for. So it doesn't give you as much information as RNA-seq, but we can actually do qPCR in our lab. It's faster, it's cheaper, so it's better if you just have a couple genes you want to look at. But with RNA-seq, we can see the expression changes of any gene in the cell, whatever we want to look at. We get the information for all of them. And then currently, what we're working on is ultra-performance liquid chromatography. This is used to analyze the presence of specific compounds in a solution. So we're using this to try to see if we can observe those metabolites after BAP metabolism. Basically, you extract the compounds of interest from the cells or media, and then you run the solution through a series of columns to separate out the compounds. Those columns can separate them by their ionization, polarization, molecular weight, molecular size, their interactions with water, any property really. And then you can identify compounds once they come out the end by their retention time. Yeah, and then our future work, we're kind of starting on this already, but it's not the priority at the moment. So first is activity-based protein profiling. We are collaborating with the Pacific Northwest National Lab, so they can do this for us. And this analyzes gene expression via functional protein. And it kind of does the same thing as qPCR and RNA-seq, except it looks at functional proteins. So you collect the total protein in the cell, you mix it with protein probes that identify specific proteins, and those probes will only connect with the proteins that are still functional. And then you can assess the protein level. So the reason we're doing ABPP over looking at all of the protein that is present in the cell is because sometimes these chemical exposures can increase the RNA and make more protein, but a lot of those proteins are actually mutated in a way to where they're no longer functional. They can no longer interact with chemicals and metabolize them. So we want to see how these PAHs are affecting the RNA expression and gene expression, and how that correlates with actual functional changes in our cell via the proteins. And then another method we're trying to incorporate is the Comet chip. So this is a new method that was recently adapted from the Comet assay, which is a very commonly used assay to assess DNA damage. So you would dose the cells, and then you would load the cells into a gel matrix. So they would be single cells in a matrix layout in gel. You would add a fluorescent compound that combines with the DNA, and then you conduct electrophoresis, so you run a current across the gel, and then you can image the cells to view what are called comet tails. So what the electrophoresis does is it breaks the cell apart to allow the DNA to move with the current. And if there has been DNA damage, then you would have small fragments and large fragments of DNA. And the more DNA damage there is, the longer the tail would be. So the smaller fragments would go really far, and the large bundles of DNA would stay kind of where the cell was originally. And then you can assess the amount of DNA damage based on those tails. So this is the results from the past work before I got here. So for all of these experiments, we are looking at 3D NHBE cells dosed with 1 to 50 micrograms per mil of BAP and PBS. And PBS is just like a saline water. It's just a vehicle to dose the cells with the BAP. And then we dose the cells for either 0 or 48 hours. And we dose them at the air-liquid interface, which is the air-exposed side of the cells. So we're trying to mimic inhalation. So if you inhale a chemical, it's going to be on the air side of the cells. And the media would just be like the interactions with the other cells in the body. So we want to mimic the inhalation as best we can. So first there was some RNA-seq data that you can see in the heat map on the right. So the green cells means that the gene was underexpressed, and the red means that it was overexpressed as compared to a vehicle control, which means that the cells were just dosed with the PBS. And the lines you can see on the left of the heat map is what's called hierarchical clustering. So this just means that the computer program we used to create the heat map found similarities in the connections that the line shows. So you can see the line on the very left connects pretty much every gene that's shown here. And all of these genes are actually related to metabolizing proteins, which is good for my thesis because it shows we are actually seeing changes from these PEHs in the gene expression levels. And I've actually boxed out CYP1A1 and 1B1. These are the two most important metabolizing enzymes related to BAP bioactivation and detoxification. So we wanted to look at those a little more closely, so we did qPCR. And we did a dose response. So RNA, we just kind of give them all the same dose so that we can see just what's happening between the different genes. But with qPCR, we can actually do a dose response. And this is like the holy grail of toxicology. You will hear dose response related to absolutely everything because it's really good for government to make regulations based off of. So you can see the mechanisms and effects from the high doses that you need to be worried about, but you can also see the low exposure amounts at the bottom. So government tries to create regulation that reduces the amount of exposure while also being cost efficient. So ideally you want exposures to be at 0, but the closer you get to 0, the more and more expensive it becomes. So the government is trying to find that sweet spot between money efficiency and low adverse effects. So that's why toxicologists love doing dose responses. And from doing this dose response, we actually see that we get a dose response of gene expression that correlates with the dose response of BAP. And what's interesting is that the low dose of BAP actually decreases the expression of both of these proteins, while the higher 2 doses actually increase the expression. And this is actually unique for each PAH. There was another student who found that if you do the same experiment with DBC, you get a completely different dose response to where I think all of them were increasing expression, and none of them were decreasing. So that's something that the grant also wants to go towards, is defining all of these characteristics for different PAHs. So the current work that I'm doing, like I said, is with UPLC. The graphs on the right are like our proof of concept graphs. So these were done again before I got here, and the extractions were done very quickly, very roughly, just so we could see if the metabolites might be there. And we actually were able to detect 2 metabolites. Yeah, 2 of the BAP metabolites in both the cells and the media. And so with this proof of concept, we now want to improve our extraction efficiency, and that's what we're currently working on. So we want to make it to where these graphs are much cleaner so that we know the peaks that we are seeing are in fact the metabolites, and there's not some strange cell protein that happens to be lingering around that's the same size and has the same retention time as the metabolites we're looking for. So this is kind of future work and also kind of what we're still working on. Like I said, the Comet chip, we're still in the method development stage. So the Comet chip was actually developed with a different cell type. It was developed with liver cells. So we have to change the protocol we were given by the company to work with our cells more efficiently. And so we're still working on that. And then the ABPP, we got our initial data from PNNL. And it showed global protein levels, so just the overall levels of proteins in the cells, that there were CYPs and GSTs that were present, but they weren't able to functionally label them. And so ABPP was actually developed using animal cells. So they did an experiment with an animal and then took like an entire organ and used that organ to do ABPP, and they got great results. But with our cell culture work, we don't have as many cells per sample. So that's something that we're trying to work on, is figuring out how many samples we need to pull together to have enough to evaluate the functional protein levels. And we don't want to just give them an infinite amount because it is expensive to do the 3D cultures. And then future goals. So something we want to work towards is creating a toxicokinetic model for BAP metabolites. And this just means that we want to be able to describe where the BAP goes. So how much is the parent left over? Does it go into the cells or the media? How much of the parent is metabolized into each of the different metabolites? So that's something we're working towards. And that's something that the UPLC data will really help with. And then we also, eventually for that third aim of the grant, we want to create a physiology-based pharmacokinetic model for pH exposures in these 3D cells. So PBPK just means that you describe, again, where the compound goes in the body. What's the exposure route? If it goes through inhalation, what organs does it get distributed to? How much goes where? How much is metabolized into what? How much is excreted? But it's not specific just for BAP. It can be adapted into all pHs that are similar to BAP, such as DBC. So that's something I really want to work on because that's getting towards computational toxicology, which is something I'm really interested in. So hopefully the project can move along and I can get to that part. So that was my research. I just wanted to give y'all some of my tips for grad school. So before grad school, before you even apply or while you're in your interviews, just some basic tips. Don't be afraid to be honest in your interviews. Like I said, I interviewed at 2 schools, and every single person that interviewed me asked, why are you leaving physics? Why are you coming to toxicology? Why should we accept you? And I was just honest. I was like, I have zero knowledge of this field. I have basically no background in biology, but I want to learn more. That's why I'm coming here. I want to learn more. And they liked it. I was accepted to Colorado and OSU. So yeah, don't be afraid to admit when you don't know something. The point of grad school is for you to learn how to learn, not for you to show off how much you learned in undergrad. And pretty much everyone understands that. So next tip is to start researching now. I was lucky enough to do research with Dr. Sanders while I was at Troy. I also did an REU at UAB. And even if you don't go into the interview process with publications, because I didn't, it still shows that you have the dedication to do research. It still shows that you are working to learn the scientific process. And even if you do come out of those situations with publications, that's like an A+. Like, schools will be fighting over you if you already have publications. And lastly, don't be afraid to try something new. Even if you know exactly what you want to do in the future, don't be afraid to take a class that's like completely irrelevant to what you want to do, because everything is interdisciplinary nowadays, and most schools want students who are interdisciplinary, because it just advances science so much faster if you can combine all of these different areas of science. And so schools really appreciate that. And then when you get to grad school, definitely connect with your cohort, which are the people who come in at the same time as you, or just the other students in your department. We have an organization called Team Tox, where all of the toxicology students get together, and we actually do outreach in the community. We do hangouts with each other, and it's great to just come together, support each other through this process, and also create a lot of friends along the way. And it helps like create a sense of home. Also, determine your own work-life balance. One of the most common interview questions, we actually just had recruitment interviews a couple weeks ago, and one of the most common questions from the students was, what's the work-life balance like? And I just want to say, do not let your job determine your work-life balance, because you will not be happy. You have to figure out how you work best. There's some students at OSU that work only Monday through Friday 9 to 5, and they're very strict about it. Their time at home is their time at home. And there's some students that just work throughout the week. They work the weekends, they work whatever hours. That's me. I just work whenever I need to get stuff done. And then there's some students that work 24/7 and they love it. So you just need to determine how you work best, how you're happiest, and don't let the job decide for you. And also ask lots of questions. Like I said, grad school is about learning how to learn. And no question is a stupid question. Like, even if you need to ask, is 2 + 2 4? Someone's going to say yes, and going to be glad you didn't write down 5, because it will mess up your experiment if you do. So don't be afraid to ask questions, even if they seem dumb. Even if you think, like, I know a lot of people watch like scientific webinars and stuff and are just afraid to ask a question because they think it's going to be irrelevant, or, oh, they already thought about it, it doesn't matter, their experiment's already done. No, like, ask the question. It may be that you have a really good insight that could help them with their next experiment or help you form your next experiment. Yeah, and so I just want to give some acknowledgements. All of my funding comes from the NIH, both through the SRP grant that we get for our lab, and also a training grant that NIH gives to OSU. And then also a shout out to PNNL for our collaborations. And I'm about to be a published author. So one of our courses, we have a special topics course, and one of our group projects was to write a vaping review. And it has been accepted for publication and should be out sometime this year. Yeah, so with that, I will take any questions. If you have a question after this, feel free to email me. My email is right there at the bottom. And yeah, thanks for having me. Not sure if I'm in the screen here. There we go.
All right. Thank you for the presentation, Tori. And I guess we will take questions. There should be a little raised hand. I don't.
Here.
So,
So you got a question, Alex?
I do.
Alex has a question.
So, Tori, you talked about like being honest with the interviews for like graduate programs. How does it feel like going through the process, like one interview? Like how many times did you interview before you got accepted?
I only interviewed at Colorado and OSU, but for each of those schools, I think I had 5 to 7 faculty interviews, and they're like 30 minutes back to back. It can be very tiring. But yeah, you just have to be ready with questions to ask the faculty. If you sit there awkwardly in silence waiting for them to ask you questions, it's going to be a little awkward. Yeah, and just plan ahead for what you think they're going to ask you. So I knew they were going to ask me why I left physics, so I was prepared to talk about that. And thankfully, there was one student that warned me about this one faculty member who is typically very tough in his interviews. And his first question to me was, "Why should we not accept you?" And I started crying on the inside. I was terrified. And then the next question was, "Why should we accept you?" So yeah, people are going to throw random questions at you. They're going to throw hard questions at you, and you just got to be very honest about why you're there, why you want to go into the field you are applying for. And yeah.
Thanks.
All right, Caroline, you have the next question.
Yeah, so you said that you're deciding between Colorado and Oregon. I was wondering what was your your decision-making factor that made you want to go with Oregon, and also how do you like living in Oregon coming from Alabama?
Yeah, those are also 2 very common questions I get. So there was a few like small factors. One thing that someone told me at Colorado was that if I went to Colorado, I had to do animal research, and I just didn't like that. At OSU, there were so many labs that did cell culture work and other non-animal research, so that was a big factor for me. Also, when I was in Colorado, I had a nosebleed for the entire time I was there because the air was so dry and my sinuses suck. So that wouldn't have been fun to be constantly sick in Colorado. Also, it was very cold. So there was just a few things. And in terms of living in Oregon, no one really believes me for some reason, but Oregon is very similar to Alabama, especially in terms of landscape. So it's really nice because like it's warm in the summer, it's still cold in the winter. It hardly ever snows, just like in Alabama. Trees everywhere, it's beautiful. Yeah, and it feels like a small town, and I came from a small town in Alabama. So it feels like home, even if I have to like call my parents to talk to them.
Okay. Is there another question from somebody?
Actually, so
You have another one?
okay. I actually enjoyed this person. I mean, I actually enjoyed Tori. So, when you do research, like especially your first year of research, how was it compared to your last year of research? Like, do you feel like you're a lot more confident with it and like go straight to work industry, or you still feel like you can still learn stuff along the way?
You definitely still have to learn stuff along the way. So all of my previous research was physics-based, dry labs, working with sound waves with Dr. Sanders, or working with crystals at UAB. So when I got to OSU, there was definitely a pretty steep learning curve because I didn't know any of the experimental protocols. I didn't know how to work with cells. But I do feel like getting some research experience, no matter what field it is, before going to grad school is definitely essential because you start that critical thinking process. So a problem pops up in cell culture like, oh, like half my cells died. Well, okay, yeah, I probably don't know why exactly, but I can think through the protocol. Where did I maybe mess up? I can think through other situations, like, oh, maybe it's because I waited slightly too long to do this thing. So it definitely helps to start getting that critical thinking gear in your brain going, and then you can apply that. Like I said, grad school is about learning how to learn. So if you can learn how to research before you start researching, that's a big help.
Thank you. Are there any other questions for Tori?
Yes, I do.
Yes.
Tori, very impressive work and great job. Did you? I feel it's a lot of biology. Did you have to take graduate level biology courses before starting this research, or you were ready to go?
No, I didn't. So I did take a biochem course at Troy, and my PI thought that that was enough to get started with my toxicology research. I did take the initiative to take a genome biology class that went further in depth on DNA replication, DNA mutation processes, stuff like that, and that's really helped me. So even though my PI didn't think I needed it, I went ahead and took a course, and it was a big help.
So it was not required?
It was not required, no.
Okay.
Okay. Do we have any other questions?
Tori, I have a question for you at the end, but can you guys hear me?
Yes.
But very good to see you. I had no doubts that you would shine, and as I see your presentation, I can see that that is exactly the case. I promise I have a question, but I would like to thank Dr. Sanders for taking the initiative to set this up. And I know the Chemistry Physics Club is also active in setting this up. To the graduating seniors I see in the participants list, this is what I expect from you guys next year. So work hard and present. Very nicely done, Tori. But my question itself for you is, Troy is a much smaller school. But what are the things we could do differently to help future researchers, future students like you?
That's a great question. I think definitely this process of having alumni talk about their grad school experience, because I know kind of the only piece of advice I was given was go to grad school. So yeah, I think this seminar series that you're all doing is going to be very helpful. I think y'all did give us a lot of information about REUs, which I think is great. Definitely keep that up. And if you're able, do more faculty research. Working with Dr. Sanders, I think, was definitely helpful.
Yes, I see that completely, yes. Again, we're smaller on instrumentation compared to a big research school. You may not know this, but we have a new polymer center with lots of instrumentation. So actually, chemists get to work with a lot of instrumentation. But will a sort of a lab sciences course for all chemistry majors separately and maybe physics majors, or a combined lab kind of course, would that be helpful with general introduction to instrumentation and things of that nature?
Yeah, I definitely think so. Also, one of my favorite things, the undergrads and students probably won't enjoy this, but one of my favorite things with grad courses is that they're mainly centered around a final project. And so even with lab courses, like you learn some instrumentation at the beginning, but then you're expected to kind of take your own initiative, find your own project, and do a final project and kind of present like a mini thesis of what you found and what you did over the course of the term. So the students might not enjoy it, but it would be super helpful if you could implement something like that.
Good point, good point. Tori, thank you very much. I won't take up your time. Like I said, very impressed, not at all surprised. And we have a good batch this year as well. So Sam, Caroline, Nick, Suraj, I see many of you here. We want to see something like this. We look forward to hearing from all of our alumni. And please stay in touch. And Tori, please stay in touch as well. Let us know of all your accomplishments. As a matter of fact, if you send me your publication list, I don't mind, as they come along, updating it in our alumni website.
That'd be awesome. Yeah.
Okay. Well, thank you.